In our conversation today, we're going to discuss the main features of biological macromolecules. And we are going to start with the general concept of a macromolecule. Macromolecules are composed from monomers, molecules that are similar to each other, and then combine with each other by covalent bonds. In that process that you can see right here, two different monomers, this and this, are combined together, releasing water. This process is called dehydration synthesis. Hydrogen of one of the monomers combines with hydroxyl group of another monomer, and two hydrogens and oxygen form water. The reason why biological systems are so versatile is because the combination of different monomers can result in production of different polymers. Not only monomers can be different, they can combine in different patterns. For instance, glucose can form three different polymers by combining in different patterns, forming different types of bonds. When a living cell has to break down polymers into monomers, it uses the reaction of hydrolysis. In reaction of hydrolysis, water is added to a polymer, and this reaction is usually catalyzed by the enzyme as well as synthesis. So water breaks down this bond, resulting in the restoration of hydroxyl group and the hydrogen on another molecule. Usually, the reactions of hydrolysis of polymers result in the release of energy. The first group of polymers, the first group of biological macromolecules are monosaccharides. The empirical formula is CH2O taken n times, and will depend how large or how small the monosaccharide is, carbohydrate. And this empirical formula represents the ratio of carbon, hydrogen, and oxygen atoms in a monosaccharide molecule. So monosaccharides are simple sugars. It means that they cannot be broken down into smaller sugars. Monosaccharides can have anywhere from 3 to 7 carbon atoms, and their names end with the suffix OSE. Depending on the position of the carbonyl group, highlighted in this image in green, monosaccharides can be aldoses and ketoses. If the carbonyl group forms aldehyde, like in glyceraldehyde, it's going to be an aldose. If the carbonyl group forms keto group, like in dihydroxyacetone, this will be a ketose. Depending on the number of carbon atoms in the monosaccharide, they can be divided into, grouped by being trioses, having 3 carbons, pentoses, having 5 carbons, hexoses, having 6 carbons. The most common, pretty much exclusively, the monosaccharides that are found in biological systems are going to be trioses, pentoses, and hexoses. Perhaps 3 most well-known hexoses among monosaccharides are glucose, galactose, and fructose. The formulas represented here, they have 6 carbons because they, hexosis, and 2 of them, specifically glucose and galactose, are aldoses, highlighted carbonyl groups, form an aldehyde. Fructose, on the other hand, is a ketose, highlighted carbonyl group, forms a keto group. Monosaccharides exist in 2 different forms. As solids, they exist as a linear chain. In the aqueous solutions, though, these linear chains fold on each other and form ring-shaped molecules. For instance, glucose can form the ring-shaped molecule either alpha or beta glucose. They differ by the position of hydroxyl group here. These are ring forms of the pentose called ribose and the hexose called fructose. If OH group is below the carbon 1 right here, we call it alpha glucose and consider that OH group to be in alpha position. If it's above, it is beta glucose because OH group is in beta position. Disaccharides are molecules that are formed by 2 monosaccharides, either same or different, and they are formed like other biological molecules in the reaction of a dehydration synthesis. In this reaction, as you may remember, the hydroxyl group of one monosaccharide combines with the hydrogen of another, forming what's called a glycosidic bond and releasing water. If the OH group is below the ring pane, in the first glucose, if this OH group is in the alpha position, we call this alpha glycosidic bond. If it is above, we call it beta glycosidic bond. The glycosidic bond is essentially an ether bond between 2 molecules of monosaccharide that comprise a disaccharide molecule. 3 disaccharides that you must know are lactose, maltose, and sucrose. Maltose, shown here, is formed by 2 glucose molecules. Lactose, shown here, is formed by glucose and galactose monomers and is naturally present in milk. Sucrose, or table sugar, can be seen in this panel, and it is formed by the combination of the glucose and fructose monomers. When there are many monosaccharides that form a long chain, we call this a polysaccharide. In a polysaccharide, individual monosaccharides are connected by different glycosidic bonds. These polysaccharides can be branched or unbranched and may even contain different types of monosaccharides. Main examples of polysaccharides are starch, which consists of amylose and amylopectin. You can see that the difference between the two mostly in the fact that amylopectin is a branched molecule, while amylose is not. Cellulose, found in plants, glycogen, found in animals, and chitin, found in exoskeleton of insects and in fungi. Starch contains, as we mentioned before, two distinct polymers: amylose, in which glucose molecules are connected by alpha 1,4 glycosidic bonds, and amylopectin, in which glucose molecules are connected either by alpha 1,4 or alpha 1,6 glycosidic bonds. That second set of bonds, alpha 1,6 glycosidic bonds, explains the branched structure of amylopectin. Bonds in glycogen are very similar to the bonds in amylopectin, while in cellulose the bonds are not alpha anymore. They are beta 1,4 glycosidic bond. You can see them here and here and here. Beta 1,4 glycosidic bonds explain the tight packaging of cellulose polymers, and that tight packaging makes them undigestible for human gut. When you eat cellulose-rich food, for example, celery, cellulose passes through the gut undigested. Finally, in chitin, the units that form the polysaccharide are N-acetyl beta-D glucosamine. We don't need to know the actual structure of these units. Next biological molecule that we're going to talk about, the category of biological molecules that we're going to talk about, are lipids. Lipids are hydrocarbons, and you may remember that hydrocarbons are hydrophobic for the most part. These lipid hydrocarbons contain mostly nonpolar bonds, such as carbon-carbon and carbon-hydrogen, which explains the lack of polarity and general hydrophobicity. We're going to start with the concept of a triglyceride, a fat molecule, or triacylglycerol, shown here. Triacylglycerol consists of a molecule of the alcohol called glycerol. It's shown on this panel, and you can see that it contains 3 carbons and 3 OH groups, and 3 molecules of fatty acids. This is the typical fatty acid, and on this illustration you can see 3 different fatty acid molecules forming ester bonds with the glycerol residue. So the length of the fatty acid chain in triglycerides or triacylglycerols can vary from 4 to 36 carbons. When there is only one, when carbons in a fatty acid molecule are connected exclusively by single bonds, we call this fatty acids saturated. It means that they are saturated with hydrogen atoms. When a fatty acid contains a double bond, we call the fatty acid unsaturated, so it can be further saturated by the addition of hydrogens. When only one double bond is present, these fatty acids are called monounsaturated, for instance, fatty acids that are found in olive oil. When there is more than one double bond, we call them polyunsaturated, for instance, in canola oil. In general, mono and polyunsaturated fatty acids are liquid at the room temperature and can be found in plants and in fish. Of course, there are exceptions. When fatty acids are saturated, they usually are solid at the room temperature, because long and straight saturated fatty acids pack very tightly. These saturated fats can be found mostly in animals. The examples of such saturated fatty acids are stearic and palmitic acids, commonly found in meat, and butyric acid, commonly found in butter. Fats for a long time were believed to be detrimental to human health, but it is not as simple. Generally speaking, consumption of saturated fats does not contribute significantly to the risk of developing cardiovascular disease. Unsaturated fats actually decrease the risk of cardiovascular disease. It's been demonstrated as well that the diet moderately rich in fats provides great health benefits in terms of the control of the body weight. However, there is a category of fats that is indeed detrimental for human health. Those are trans fats. So, plant oils, plant triglycerides that we call oils, are much cheaper and easier to obtain. These oils can be hydrogenated, in other words, hydrogen can be bubbled through them, to convert them to saturated fats, semi-solid fats, such as margarine or vegetable shortening. However, during the hydrogenation process, unsaturated fatty acids that normally have cis conformation of a double bond change its conformation to trans. You can see the difference between cis and transoleic acid. Cisoleic acid molecule is kinked, which explains why these molecules cannot be tightly packaged, therefore they will form liquid oil. Transoleic acid, on the other hand, is a long straight molecule. These molecules can stack very tightly, and that explains why trans fats are usually semi-solid or solid. Turns out that when hydrogens are located in the same plane and they form regular liquid oils, cis fat, they are good for the cardiovascular health. Hydrogens into different planes, trans fat, semi-solid, actually significantly increase the risk of atherosclerosis and heart disease. These trans fats specifically raise the level of what we call low-density lipoproteins, LDL, or bad cholesterol. So when you buy foods, make sure that it does not contain trans fats. Some fatty acids cannot be produced inside of the human body. They still required but can be synthesized. The categories of those fatty acids, two most common categories of fatty acids, are omega-3 and omega-6 fatty acids. These are found in salmon, trout, and tuna. So why are they called omega-3? If you would look at this panel, this is the last carbon atom in the fatty acid. The last letter of the Greek alphabet is omega. So if we start counting from here, the position of the double bond is at 1, 2, third carbon. We keep counting, and next double bond is at 6. So it's going to be 3, 6, so omega 3,6 fatty acid. It's been demonstrated that fatty acids may reduce the risk of heart disease. They may reduce the levels of triglycerides, fats in the blood, positively influence blood pressure, and prevent thrombosis. They may even reduce the level of inflammation and lower the risk of cancer in animal models. Another type of lipids that we need to talk about are waxes. Waxes are formed by fatty acid chains that are esterified by a long chain alcohol. Waxes, due to the really long hydrocarbon chains shown here as a zigzag line, are extremely hydrophobic. When they cover a plant leaf surface, they prevent water from sticking to it. When they cover the feathers of aquatic birds, they carry out the same function. So feathers of birds and plants' leaves are two main locations for the waxes. Next category of lipids are phospholipids. Phospholipids can be found in plasma membranes and in membranes of the membrane-bound organelles in eukaryotic cells. So phospholipids are formed by fatty acid chains that are attached to a molecule of glycerol, shown here and here, or the molecule of sphingosine. Additionally, glycerol or sphingosine have the phosphate group that carries some kind of a modification, some kind of a radical. This radical, shown here as R, is usually an alcohol. The examples of phospholipids, shown on the right, are phosphatidylcholine as a modification and phosphatidylserine as the modification. If you would look at the molecule as it is represented on the left, you may notice there is a negative charge on the phosphate group. There are a lot of very electronegative oxygen atoms here, which makes the head hydrophilic. The tails of phospholipid molecules are comprised of fatty acids, and that makes them hydrophobic. This is why phospholipids are often pictured having hydrophilic head and hydrophobic tails. So that feature, having hydrophobic part and hydrophilic part, is called being amphipathic. So phospholipids are indeed amphipathic. These hydrophobic tails that are composed of fatty acid chains can't interact with water since they're hydrophobic. On the other hand, the head, which is hydrophilic, the head that contains the phosphate and the glycerol, actively interacts with water. This dual function, or should I say dual functionality of these molecules, makes them ideal to form a membrane. Bilayer of phospholipids have hydrophilic surfaces facing the extracellular environment and intracellular milieu. Hydrophobic tails form very selectively permeable barrier in the middle. If you would take phospholipids and drop them in the water solution, it will spontaneously form a structure called the mycele. You can see that in the mycele, the tails, composed by hydrophobic fatty acids, they face the inside of the mycele. The heads, on the other hand, the polar heads, they face the outer environment, which is, as you remember, water. The last type of lipids that we're going to talk about are steroids and hopanoids. Steroids, two examples of which are seen on the right, cholesterol and cortisol, have the structure composed of the fused rings. Steroids also are hydrophobic, lipids are, and are insoluble in water. All of them have 4 linked hydrocarbon rings, 1, 2, 3, and 4. Some of them, however, are modified. For example, cholesterol has this hydrocarbon tail. Many of them have functional groups, such as hydroxyl group, methyl group, carbonyl groups. And they serve in different capacities in human and other animal cells. Cholesterol, for example, is the molecule which is used to form many steroid hormones, such as testosterone, estradiol, or aldosterone. It is also a precursor for the vitamin D and the bile salts, as well as being precursors in biological reactions. Sterols regulate the rigidity of plasma membranes, specifically in the eukaryotic cells. In bacteria, there are similar structures called hopanoids, for example, hapine at the bottom, that regulate the rigidity of the membrane. As you can see, hopanoids contain not 4, but 5 fused carbon rings. Our next biological macromolecule is proteins. Probably the most abundant organic molecule in biological systems, which has the most versatile and diverse range of functions. They can serve in the structural capacity, forming various structures such as cytoskeleton. They can be regulatory, for example, hormones, to coordinate the activity of different systems in the body. They can provide the ability to contract, to giving cells the ability to move. They can serve to protect, such as immunoglobulins that protect human body, and not only human body, but animals against various pathogens, serve as antibodies. They can transport, for instance, hemoglobin and albumin can carry substances in the blood or lymph. They can provide storage. Egg white or albumin stores nourishment for the embryo development, or storage proteins of legumes provide the nutrients for the development of the seedling. They can break down other molecules, serving as digestive enzymes. They can break down food. As a matter of fact, most of the enzymes, biological catalysts, are proteins. In addition to that, they can be parts of the cellular membrane, or they can serve as toxins, especially in bacteria. The structure of the protein determines its function. Since structures vary greatly, the functions can vary greatly as well. The elementary building block of any protein is amino acids that are initially arranged in the linear sequence. Amino acids, monomers that comprise proteins, share same fundamental principal structure. The central atom called alpha carbon is connected to an amino group and a carboxyl group. These three, this whole arrangement that I just highlighted is pretty much standard for all amino acids. What makes them different is the presence of the side chain or the R group. 20 different common side chains form 20 different common amino acids and proteins. None of them are essential, meaning that we humans can't produce them and have to get them from the diet. Different R groups of different amino acids determine essentially their chemical properties. Amino acids can be chemically nonpolar, for instance, valine, can be polar, for instance, serine, can be charged, such as lysine or glutamate. When protein sequence is written, amino acids usually represented by an uppercase letter or three-letter abbreviation. These letters and abbreviations are unique for a specific amino acid. When two amino acids react with each other, combining a carboxyl group of one and an amino group of another, they form a peptide bond. This is another example of dehydration synthesis because in this process water is released. This covalent bond between two adjacent amino acids is the same between any two adjacent amino acids. Essentially, the sequence of amino acids and the number of amino acids in a polypeptide determine the structure and the function of the protein. Here you can see different amino acids based on the charge, polarity, or not polarity. So you should be able to distinguish, should be able to identify based on the structure amino acids as nonpolar aliphatic, nonpolar aromatic, polar uncharged, negatively or positively charged amino acids. If you look at nonpolar aliphatic, you can see that there are no, they only contain basically carbon and hydrogen in the side chains, well, except for a little bit, except for methionine, which contains sulfur, and they do not have aromatic rings. Nonpolar aromatic amino acids contain aromatic rings, such as phenylalanine or tyrosine or tryptophan, which is aromatic heterocyclic. Polar and uncharged amino acids contain some kind of very electronegative atom, such as sulfur, oxygen, and this oxygen can be in the form of OH group, hydroxyl group, or in the form of carbonyl group. Positively charged amino acids obviously carry a positive charge, usually on a nitrogen. Negatively charged amino acids carry negative charge on the, should remove this one, carry negative charge on the carboxyl group in the side chain. The unique sequence of amino acids in a polypeptide chain, as the sequence shown on this illustration, sequence of amino acids in the molecule of insulin, is called its primary structure. Every protein is encoded by a gene. The sequence of nucleotides in a gene determines the sequence of amino acids in a protein. If this gene is changed, for instance, one nucleotide is replaced by another, this replacement, that we call a mutation, may lead to a change in the sequence of amino acid in the protein encoded by that gene. This change may, sometimes may not, alter the structure and therefore the function of the protein encoded by that gene. interactions between oxygen atom in the carbonyl group of one amino acid and the hydrogen atom in the NH group of another amino acid facilitates a folding of polypeptide, essentially forming its secondary structure. So, secondary structure is the local folding of polypeptide chain in some of its regions. Two most common structures that you need to know that represent secondary structure of a protein are alpha helices and beta pleated sheets. So you can see alpha helix on top and beta sheet on the bottom. So when alpha helix is formed, R groups, side chains, protrude out of the helix and extend above and below. In the beta pleated sheets, side chains stick up or stick down. In beta pleated sheets, the strands can be either parallel or anti-parallel to each other, as you can see in this particular example. They are going in two adjacent beta strands, go into opposite directions. The interactions between side chains of amino acids form its tertiary structure. These interactions, as a matter of fact, can counteract the structures formed at the level of secondary structure. How does that happen? Well, for instance, if adjacent R groups carry the same charges, they will repel each other. If they carry opposite charges, they will be attracted to each other. For instance, if you look at this, the positive charge on the amino group and negative charge on the carboxyl group facilitates the formation of ionic interaction. Hydrophobic side chains can interact and be attracted to each other due to the hydrophobic interactions. Partially positive hydrogen and partially negative oxygen of two different side chains can result in the formation of hydrogen bond. And sulfhydryl groups of two adjacent cysteine molecules within the polypeptide can form a covalent disulfide linkage. This is the only covalent bond that is formed during the folding of the protein. Now, if you would look at the tertiary structure of the protein, you will quickly recognize that side chains that are hydrophilic face the outside of the protein because the intracellular or extracellular environment is hydrophilic. On the other hand, within the protein molecule, the R groups are mostly hydrophobic, mostly nonpolar, so they don't have to be repelled with the water. It's important to understand that when all these interactions are interrupted, the protein loses its structure and therefore loses its function. This process of a protein losing its structure and function is called denaturation. In case of some proteins, several polypeptides or subunits are required to form a functional molecule. Interaction between these several subunits, for instance in a molecule of beta globin, such as hemoglobin, is called a quaternary structure, and usually quaternary structures are stabilized by the combined action of hydrogen bonds and disulfide bonds. A perfect example of what can happen if one amino acid is changed is a disease called sickle cell anemia. In sickle cell anemia, there is a single substitution of amino acid in the position 6 of a hemoglobin molecule. The polar glutamate is replaced, actually yes, the polar glutamate is replaced by an unpolar amino acid, valine. That single amino acid change completely alters the structure and the function of this protein. Consider that there are 600 amino acids in a hemoglobin molecule. What happens is that this, instead of forming globules, normally, hemoglobin molecules start forming fibers. These aggregated fibers change the shape of the red blood cells from biconcave to a crescent or sickle. This results in hemolysis, kidney failure, shortness of breath due to the lack of proper oxygen transport, dizziness, headaches, bleedings, abdominal pain. This disorder is inherited. So mutation goes from parents to kids. It is a recessive, autosomal recessive disorder. So in order to develop sickle cell anemia, a child has to inherit sickle cell genes from both their father and their mother. The final type of biological molecules that we're going to talk about today is nucleic acids. There are two main types in the cell: deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. DNA serves as the genetic material in all living organisms. That includes anything from bacteria to fish to insects to plants to fungi to mammals. Quite literally, if you look at the genome of any living thing, any living cell, this genome will be formed by the molecule of DNA. In eukaryotic cells, DNA is complexed with special proteins called histones. This complex between DNA and the histones is called chromatin. Chromatin eventually can condense to form chromosomes. Each chromosome contains tens of thousands of genes that contain the information allowing cell to produce proteins, or RNA. In eukaryotes, DNA, the genomic DNA, is located in the nucleus, while extrachromosomal DNA can be found in mitochondria, and if we're talking about plants, for example, in chloroplasts. As we mentioned, it functions as genetic material. In prokaryotes, DNA can be found in a nucleoid. Nucleoid basically is the DNA bound to special proteins, but there is no membrane that surrounds nucleoid. It also functions as a genetic material. You have to remember that DNA controls all the cellular activities, and it can be done by turning the expression of the genes on or off. Another critical molecule here is RNA, or ribonucleic acid. It immediately controls protein synthesis. The most important intermediary here is the so-called messenger RNA. Other types of RNA, such as ribosomal, transferred, microRNA, or small interfering RNA, are also involved in protein synthesis and its regulation. In eukaryotic cells, RNA can be found in the nucleus and in the cytoplasm, and as we mentioned, it is a critical molecule for the gene expression. In prokaryotes, it can be found in the cytoplasm, since they don't have a nucleus, also serves as the gene expression intermediary. I want to highlight that in eukaryotic cells, DNA can be found only in the nucleus or mitochondrial chloroplasts, but not in the cytoplasm. Now, I apologize for that long pause. Everything's fine. It's just my computer got a little frozen. So I want to reiterate the following, that in animal cells, DNA can be found only in the nucleus, but not in the cytoplasm. So now let's take a look at the actual function of DNA, RNA, and protein in the context of gene expression. DNA, essentially, has all the instructions for protein or RNA synthesis. I call it the recipe book. You don't carry the entire recipe book to the kitchen. You just take a little picture, a little photo of the recipe that you need to produce. RNA serves as the photo of the recipe. This photo of the recipe carries the instructions to the kitchen of the cell, a ribosome, which uses the instructions from RNA to produce a meal, an actual protein or another RNA. If your friends are coming over to visit you, you're not going to feed them with the recipe book or pictures. You're not going to feed them with the recipe book or pictures of the recipes. You want to provide them a meal, a protein, since the majority of cellular functions are carried out by proteins. Genes have to be expressed. When DNA is used to synthesize RNA, call this process a transcription. In our analogy, it's the process of taking a photo of a recipe. When RNA is used as the instructions for protein synthesis for the meal preparation, we call it translation. So translation is essentially preparing the meal. This process is called gene expression. It's important to understand that gene expression can change in response to different environmental conditions or environmental activities. This explains why different human cells have identical genomes but look and work differently. Simply, different cells have different patterns of gene expression. Therefore, the phenotype is determined by the set of genes that are expressed at any given moment. The building blocks of nucleic acids are nucleotides. Both DNA and RNA consist of these types of monomers. However, there are slight differences between certain elements of these monomers between DNA and RNA. Every nucleotide consists of a nitrogenous base, a 5-carbon sugar, pentose, either deoxyribose or ribose, and a phosphate group that is attached to a 5' carbon. Nitrogenous base is attached to 1' carbon of a sugar molecule, whether it is deoxyribose or ribose, and the phosphate is also attached to the sugar. Within the rings of nitrogenous bases, you can find both carbon and nitrogen. You can also find amino and carbonyl groups. Nucleotides differ due to the different nitrogenous bases. In DNA, there are 4 possible nitrogenous bases: adenine, guanine, cytosine, and thymine. Instead of thymine in RNA, uracil serves as a 4th nitrogenous base. You can see that all nitrogenous bases can be classified into 2 large categories: purines, having 2 rings, and pyrimidines, having 1 ring. Adenine and guanine are purines. Cytosine, thymine, and uracil are pyrimidines. A pentose sugar that can be found in nucleic acids can be either deoxyribose if it's DNA or ribose if it's RNA. The difference is that in ribose, there's a hydroxyl group at the 2' carbon, while in the DNA, instead of hydroxyl group at the 2' carbon, there is a hydrogen atom. The carbon atoms in the pentose sugar are numbered 1', 2', 3', 4', and 5'. You may ask why? Because the numbers like 1, 2, 3, and 4, and 5 and 6 are reserved for the rings in the nitrogenous base. Now, the importance of the phosphate group attached to the 5' carbon of a pentose is that it can connect OH group at the 5' carbon, this one, of one sugar, to the OH group at the 3' carbon of another sugar. This connection is called 5', 3' phosphodiester bond or linkage, and this connection determines the directionality of nucleic acids. It's important to remember that this is not a reaction of dehydration. DNA molecule, as you are probably aware, has a double helix structure. This double helix structure is shown here. In this double helix structure, the sugar and the phosphate form the backbone. Here I highlight the backbone. The nitrogenous bases are inside, facing each other, forming some sort of a staircase. The hydrogen bonds hold the strands together. These hydrogen bonds are formed by the interaction between nitrogen, oxygen, and hydrogen atoms in the opposing bases. It's interesting to see that a purine always forms a pair with thymine, and these pairs, in fact, are fairly rigid. A pairs with T and G pairs with C. AT pair in this case is weaker than the GC pair. If you look at the image, you can see that AT pair on top forms two hydrogen bonds, while the GC pair, again, sorry for that mishap, start over. AT pair on top forms two hydrogen bonds. While the GC pair on the bottom forms three. This is GC pair. Strands of DNA in the double helix are antiparallel. It means that these strands run in the opposite directions, like this. The complementarity also allows us to figure out the sequence of one strand if we know the sequence of another. If you look at the example in the notes that I provide, you can see that if the sequence of that strand is AATTGGCC, then the sequence of the opposite strand will be TTAACCGG, basically complementary strands. Every time the cell divides, DNA is replicated. During the replication, each strand is copied independently. We call it a semi-conservative replication. Essentially, if we look at the process of replication, we can illustrate it as follows. So red color here represents the original DNA molecule. Each strand will be individually copied, and now each of the new double-stranded DNA molecules contains one strand from the original molecule and one strand that have been freshly synthesized. It's important to highlight, and you can see that strands have to be separated. So in order to separate the strands during the processes of transcription or replication, the hydrogen bonds between bases have to be broken. Breaking those bonds allows strands to separate, which exposes, we call it denaturation. The denaturation exposes nitrogenous bases, and that allows enzymes of replication or enzymes of transcription to read the sequence of nitrogenous bases in the DNA. We mentioned before that a phosphate group forms a connection between two adjacent nucleotides called phosphodiester linkage. Turns out that makes molecules of DNA directional. It can be illustrated as follows. In the double-stranded DNA, one strand runs in like 5 to 3 prime from left to right. In another one, it runs in the opposite direction. The directionality is important for the DNA function because it allows it to carry information. Very much like when you read the book, you read from left to right. You know what the text is going to be. You know you can receive information because you know the directionality of the text. Obviously, in other languages, the direction can be different, but my point is that you are always reading in English from left to right. The same way, every DNA molecule is always read from 3 prime to 5 prime end. Finally, we're going to talk about RNA. So RNA molecules are usually single-stranded. There are some exceptions that we're not going to talk about. They comprised of ribonucleotides that contain ribose instead of deoxyribose and uracil instead of thymine. Just like in DNA, ribonucleotides are connected to each other by phosphodiester bonds. As we mentioned, each ribonucleotide contains the pentose sugar ribose, nitrogenous base, which can be A, U, G, or C. And in terms of pairing, A pairs with U instead of T, and G pairs with C. And finally, the phosphate. Just like thymine, uracil, its replacement, is a pyrimidine nitrogenous base. 4 types of RNA that you must know are messenger, ribosomal, transfer, and micro. Messenger RNA, also known as mRNA, is that photo of the recipe that carries the genetic message from DNA to a ribosome. Essentially, the gene for a specific protein is turned on, is being expressed, and then when it is being expressed, the messenger RNA is synthesized in the nucleus. Ribosomal RNA represents the major catalytic component of ribosomes. It plays a critical role in aligning mRNA and ribosomes properly and in formation of the peptide bond during the protein synthesis. In this illustration, you can see mRNA on the bottom and ribosomal RNA somewhere in the ribosome. Transfer RNA or tRNA carries the amino acid. Here's the amino acid. This is tRNA. Carries the amino acid to the site of the protein synthesis. Lastly, microRNA or miRNA can regulate gene expression, can regulate protein synthesis by selectively controlling the degradation of the genetic messages in the form of messenger mRNA. Next time, we're going to start talking about a cell.